I come from the front lines of a war, and it's a war on disease. Very different from the war we read about every day in the newspaper. I'm going to talk to you today about just some of my views on this war and end up with some ideas that I hope can be a new way of thinking about these diseases. And it all comes from the fundamental, unfortunate truth that we're not much better at treating disease than we were 50 years ago. The death rate from cancer today is no different than it was in 1950. And so unfortunately, we've taken a wrong turn. And so I'm going to talk to you about a dream, and that's a dream that everyone in the healthcare community has. That is, a patient comes in, and we could do a profiling and be able to tell that patient, you need drug X, or you need drug Y, or you need surgery, or unfortunately, we have nothing for you. Remember, the unfortunate truth is, if you look at what happened in Washington over this last year, we didn't have healthcare reform. We had healthcare finance reform. And between you and I, as a patient, as a doctor, I really don't care that much about healthcare finance. I care about those two patients of mine that died this morning. And I need true healthcare reform in that regard. So the reality is that we are the #1 spender in healthcare. And we spend almost twofold more than the #3 spender. And we're ranked in the 30s in healthcare in the world. So obviously, there's something incongruous there. You take into that the problems with healthcare finance, some of which are going to be addressed by these bills, and it certainly will make the field move forward. And we have to reinvent ourselves. We have to think differently. You look at data like the cost for procedures in the United States versus other places in the world. If you take a procedure like a hip replacement, that costs or is charged on average close to $50,000 in the United States, but it's less than $10,000 in most other countries. If you go to a hospital in India, they're verticals, which means they do one thing, and they do that damn well. And so this notion of traveling for medicine is real. If you go to a hospital that does cataract surgery, that doctor is doing one patient, another, and they just add patients in as that doctor goes around. And in surgical procedures, in many of the things we do in medicine, quantity equals quality. In the United States, every hospital is incentivized to do everything. So by definition, we're relatively inefficient. Then you look at these figures, and these are figures for Canada, which the data are that we are 25-fold more than the figures shown here. But if you look at the average doctor who is seeing about 5,000 visits a year, he or she are creating 300 million pieces of paper. You take that and you multiply that by what's happening in the United States, and it's obviously a staggering waste of trees. But most importantly, we don't learn from a single of these events. In the United States, we are so good at reporting the negative. If I have a patient who goes on a drug, I have to report to the FDA every major side effect. There's no place I have to report the positive. So we don't get better in what we do at all. And so one of the things that's happening now is a trend toward healthcare reform, which is electronic health records. The power of electronic health records is we can learn. I went into a friend of mine's office. He's CEO of a company called Activision, a large computer game company. He can go into any cash register in any Walmart and see what people are buying with his product. He could change that placement of his product and make it near Tide or near Dove soap and see how it affects the sales of his product. And he could do this in real time. I can't tell you the outcome of my patients over the last several years. I learn in that my art form gets better, but I can't translate that to other doctors, unfortunately. And hopefully that trend will change. And I don't mean to be all negative. I mean, this is worldwide data on life expectancy growth. So you look today where the data, we're living to the mid-60s, and this is worldwide. This is taking sub-Saharan Africa, this is taking Asia, the United States, all together, we're in the mid-60s. And you look where we were in 1950, that is a dramatic growth from 50 to over 70 years in that time course. And so certainly we're getting better at what we do, but there's a lot more we have to improve. You know, the problem is that we do things on a disease model. You know, in the 1850s, the French came up with a classification for cancer based on body part. breast, lung, prostate, etc. The way a pathologist recognizes disease is pattern recognition. This is a normal cell, and this is a cancer cell. And that's it. He or she looks under a microscope, and they say, this looks similar to normal, or this looks similar to a disease. There's no molecular technology. We don't understand the pathology of the disease, the biology of the disease. We do a simple pattern recognition. So then the medical field went to another extreme. We went to reductionist point of view, in that the goal over the last decade was sequence every individual gene. Can we get down to understand the fundamental genetic component of a disease? And we did that. The problem is the average cancer, for example, has over 100 mutations. Can you comprehend that? Can you put that into an order? And the answer probably is no. But in nature, there are a lot of forms that go from chaos to order. You know, if I took an egg from the kitchen and I put it in this room and came back 3 weeks later, I'd come back with a rotten egg. But if I take that same egg and I put it in the room and I change it to 99.5 degrees, plus or minus a half a degree, and I rotate it 3 times a day, I have a chicken. If I rotate it 4 times a day, I have a deformed chicken. And so gravity and temperature can take something from a rotten egg to one of the most complex organisms in the world today. So obviously, small changes can make a difference. And the goal of what I'm going to show you in a few minutes is to understand and comprehend those changes. But our field has been fooled. You know, in the era or the mid-1900s, we started to have antibiotics for infectious disease. And infectious disease fooled us. Those organisms, you identify the organism, we can identify the treatment. It is very simple. We can look for a defect. If they have a cell wall, well, we give them a drug that inhibits cell wall formation and the organism dies, because the human cells don't have cell walls. We were fooled by that. And what I'll argue for you today is the way to view disease is not a reductionist like we did in the case of infectious disease, but it's to view it as a complex system. You know, Galileo used to go out every night and look at the stars. And after 4 months, he could tell you what star was where in the sky at what time of night. Well, Galileo didn't even know what a star was. My field, the healthcare field, has been trying to understand disease to the reductionist point of view, to the individual molecule. And we forgot to take that step back to look at the whole system. And I'll show you how that has come about. Remember, engineers, which are remarkable thinkers in their own field, engineers will tell you they don't have to understand a complex emergent system to control it. So my goal is not necessarily to understand cancer, it's to control cancer. You look at the majority of the grants out there by the National Institute of Health, and I'm guilty as anybody else, and they are to understand the fundamental natures, and we forget to take that control step back. Because cancer is not a disease of the genome. If you look at nature, evolution doesn't select for genotype, it selects for phenotype. If a pathologist looks under a microscope and they look on the right there, that looks like cancer of the liver. If a radiologist looks at a CAT scan, and this is a section through the liver like this, that looks like metastasis or cancer in the liver. It's pattern recognition. Evolution is selecting out for these patterns. Remember, every cancer of the liver looks like that. You don't have to go to the individual gene to figure out that it is a cancer of the liver. And so it's the same along the lines of evolution. And we fought back the other way, all on the understanding side, and we didn't look at that system side. And I'll show you how we're starting to do that and how it's starting to transform medicine going forward. And again, I don't mean to denigrate things like DNA sequencing. I'm as guilty as anybody else. And I'll show you now some technology that we've developed that has actually helped us on the medicine front using this reductionist point of view in a sense. But if you look in the Monsanto annual report in 1974, this is Monsanto, the large conglomerate, they estimated $150 million to sequence one gene. Well, today in my laboratory, it costs on the order of about 50 cents to sequence a gene. When I go home at night, I can come back with a patient sample, and I could have seen that patient that morning, and I can have 2,000 genes sampled for several dollars, and I have that data to better treat or understand the patient. And so I founded a company 3 years ago now, called Navigenics. And the goal is to improve healthcare by understanding some of the genetic risk factors. You know, I'm a cancer doc, and I realize that we are not very good at treating cancer, and we're not going to be in the foreseeable future. It took us 50 years to get where we are, and we haven't moved that far. So the goal is going to be on the prevention side. If you look at most of healthcare, it's spent on the treatment side. Remember, about 40% of healthcare dollars are spent in the last 2 years of life. We spend very little in the upfront setting. And so if we start to prevent disease, we can actually have a big impact. And so if you look at diseases like heart disease, cancer, stroke, those are in general are preventable or delayable diseases. There are 3 billion letters in the human genetic code. There have been studies done over the last several decades that start to associate different DNA changes with disease. And so the technology to do that, as we alluded to before, has gotten much cheaper and much more scalable. So if you look at the number of publications in good journals that have shown these associations, it went from a couple in 2006 to exploding in 2010 with over 200 publications of gene association studies. And so this is the first time I've showed this publicly, this is me. These are my genes, and this is telling me my likelihood of multiple different diseases. So I look at this, and you know, the national risk, for example, in a male for heart disease is 42%. My likelihood in my lifetime is 46% of having a heart attack. So because of that, I'm lean body mass, I exercise, I take a statin, I've reduced that number tremendously. And the company Navigenics has started this and is doing thousands a week of individuals in order to better understand their genome. You spit into a tube, you mail it in, and then a week later, you go online with a genetic counselor and you learn about you. It's interesting, when I put it together, I meant it for the consumer. I meant to democratize what we have in the laboratory and then what you read about in the paper every day and make it available to all of us. In reality, the majority users of this are corporations. Microsoft, Cisco, Intel, they're doing it for their employees. They realize the cost benefits here, that if they can delay disease by a decade, they've dramatically affected their bottom line. They get back a readout, not of the individual employer. What they get back is 8% of their patients will get breast cancer, for example, 20% diabetes, 10% heart disease. They know where to put their philanthropy. They know where to put their prevention. And in that sense, it makes a difference. President Bush, during his presidency, passed a law, one of the few things on the positive side, called GINA, the Genetic Non-Discrimination Act. Well, he did something else positive, is that he tried to outlaw stem cell research. The reason it was positive is that everyone in this room knows all about it now. California passed a $3 billion initiative in order to do it. So that ignorance actually stimulated a lot going on in our field. So the Genetic Non-Discrimination Act allows us, so it's illegal for an employer to make a decision based on your genetics. If you have a risk for breast cancer and they find out by some means, they can't do anything to terminate you or change it. And so what this does is it tells you your risk versus the population. Right now in our country, we do colonoscopy at age 50. Right? That's when the risks, the benefit, the costs all come together. What do you say to the 4,100 people last year that died of colon cancer under the age of 50? Well, you didn't fit those risk-reward curves. Now we have a technology that can say when you should have screening and how it should be done. I want to switch gear now and talk again about what's coming next. So what we and others are doing, I'm working on a field called proteomics. So genomics, the study of the gene, proteomics, the study of the protein. What we do is we take a drop of blood, and this goes through a superconducting magnet. So these are magnets that are 10 times larger than an MRI. And what it does is it gives us a 40 or 50 gigabyte picture of all the proteins in the blood. So this is a computer hard drive, or the old computer hard drives, now they're much bigger, of all the proteins in the blood. We can actually visualize a neutron with this. And with this, we can get a picture of your state, what's happening in your disease. and we create these large databases where we correlate what happens with what's present on the proteins. So for example, if 100 women have breast cancer, I give them a drug, 50 respond, 50 don't respond, that 101st patient, I say, is the pattern of their proteins like the responder or the non-responder? So this is an example of a woman with lung cancer. She's 46. She gotten every chemotherapy, every radiation therapy we could give her. She had 3 kids under the age of 10. And this is a section through her chest like this with a CAT scan. And you see on the left there in the red is the large mass. That's the tumor in her chest. I give her a pill and the tumor goes away. This is rare. This is very rare. The problem is that same pill in a randomized trial showed that 70% of the non-responders lived much longer. So this pill slowed the rate of growth of cancer. It didn't shrink it in most cases. And these technologies will yield the personalized medicine of the future. And this is going to happen over the next several years. So I just want to talk for a minute about that whole concept of complex system. A clinical trial was published last year where they took women with breast cancer after chemotherapy and they randomized them to placebo or a drug that builds bone. And what they found is a drug that builds bone that didn't touch the cancer reduced recurrence by over 30%, more than chemotherapy. So the notion you change the soil, the seed doesn't grow as well is real. You start to change those systems in the right direction, we're going to have a benefit for disease. And again, we've been thinking too broad. The problem is all our data sets are different and wrong because obesity wasn't really that much of a problem a decade ago. Now 3/4 of our country is approaching obesity. So our system changes. Our diseases are changing. How we respond to diseases are changing. And obviously, we have to do that. We have to develop technology to look at the whole. We have to develop a technology not necessarily to treat for shrinkage on a CAT scan, but to go from a disease state to a healthy state. So instead of a pharmaceutical company or biotechnology company developing a drug that targets a gene with the metric of making that cancer a little smaller, wouldn't it be nice if we were able to treat to go from disease state to health state? If we had a monitor for health state, and that's certainly what we're trying to do with the proteomics. And everyone always asks me, what's next? Well, I think the next stage after this is going to be something called microbiomics. The microbiome are the bacteria in your GI tract. So all of us have slightly different bacteria, and they number at 10 to 1 bacteria to human cells in the body. So they're a large part of us. They metabolize all our food. They change what we absorb and how we absorb it. So for example, if you look at the incidence of prostate cancer in the United States versus China, there's a 90-fold difference. If someone from China moves to the United States, they start to approach us in terms of risk of prostate cancer after a decade. So everyone says, well, it's because of McDonald's. It's because we're all eating poorly, because we have a high-fat diet. Well, they're starting to have that high-fat diet there, and they're not seeing these rates. Part of it is the microbiome is different there than it is here. So that next generation of technology will be able to sequence all the bacteria in our gut and know how you and I metabolize food differently. And it's going to say, listen, based on your genetics, you have a high risk of prostate cancer, so you need to have this mix of bacteria in your GI tract. And this isn't going to be science fiction. This is going to happen over the next several years. So to end, Al Gore told me this story that when President John F. Kennedy announced that we're going to land on the moon, everyone thought he was crazy. 9 years later, the headlines of the paper was, we landed on the moon, and potentially we actually saw the pictures of it if that was real. The average age in mission control was 28 years of age. So the generation of people who created us landing on the moon at the time were 19. So it ain't going to be me who make all these big changes in the healthcare. It's going to be you guys in the audience. You are the next generation that are going to develop and apply these technologies that will hopefully dramatically change disease. So thank you.